My name's David James. I'm a professor at the Charles Perkins Centre at the University of Sydney, and I am a basic researcher interested in metabolic disease. I want to understand the cause of metabolic disease, and I want to ultimately shut it down and stop it from occurring and eradicating it from the planet. I've been researching metabolic diseases, so that's things like type 2 diabetes or cardiovascular disease, pretty much my entire career. But I do so from a very fundamental point of view. So at the heart of many of these diseases is a phenomenon called insulin resistance. So this is one of the earliest defects that occurs in all of these diseases. And so my laboratory is ultimately really interested in understanding how does insulin work normally and how does its actions go awry in the disease state. So the idea is if we can understand these very early defects, this will help us provide cures or other things that could stop the disease from ever occurring. In researching complex diseases like diabetes or cardiovascular disease, even cancer, I think what we've come to realise is that for too long we've been the drunk guy stumbling home from the pub that's just lost his wallet. What we've been doing for the last 50 years is we've been trying to look for the cure for these diseases under the lamppost. And, you know, it's no wonder that we haven't actually really, I think, reached the full potential, I think, of what we ultimately would have wanted to. And so the idea with big data is we make no assumptions about where the defect is, because we don't know where the defect is. So what we do now is we just look at complex systems like humans or animals, and we collect as much data about those systems as we can, and then from the data we try to find things that predict outcomes, like whether you're going to get complex disease or not. So in my view, this is a completely different way of doing science. You know, when I was a student, I was taught you have to have a hypothesis, hypothesis-driven science. If you didn't have a hypothesis, you were never going to get the money. This new way of doing science is there's no hypothesis. You've got a problem, you collect as much data, and if anything, you use the data to find a hypothesis. I love this way of doing science. It's opened my eyes. For the last 30 years, my lab has been trying to understand how insulin works. And for a lot of that time, like many, many other labs throughout the world who are studying the same problem, we've been beavering away, focusing on individual molecules, thinking that this was going to help us understand how insulin works. So about 5 years or so ago, my laboratory used a new technology, this technology called mass spectrometry, to study a process called protein phosphorylation. So when you add a phosphate group to a protein, and the phosphate comes from ATP, I think many people know what ATP is. One of the major functions of ATP is to donate phosphate to proteins, and that changes the activity of the protein. So by studying protein phosphorylation on a global scale, we can understand all the things that are going on inside cells and tissues. We applied this technology to adipocytes. These are the fat cells in us that we know play a major part in how insulin works every time we eat a meal. And we studied protein phosphorylation on a global scale, and extraordinarily what we found is that within 5 minutes of adding insulin to those cells, there was a change in about 2,000 to 3,000 phosphorylation events in that cell. That is how insulin works. And that made me realise we need to take a step back and we need to think globally. That data set, which we published in 2013, I go back to constantly because it's invaluable. There are some gems in there. We haven't even touched the surface. It's actually relatively simple to create the data. A lot of people don't realise that. That's the easy part. The hard part is to make sense of the data. What does it mean? What's it telling you? And I've come to realise that what is so important here is to display the data, visualise the data in a way that you can actually show it to other people so that they can understand what you think the data is telling you. So we've actually spent a lot of effort on finding new ways to visualise complicated data. And one of the ways that I love, we've got a few, but my favourite one is that it's a form of virtual reality. This is it's sort of a dynamic way, an interactive way of displaying data. I think one of the problems with many visualizations is that they're two-dimensional, whereas data, the sort of data we acquire, is dynamic. It's changing in time. And so this visualization is actually living, and you can actually go in there and see things changing, and you can almost interact with it. And I think this is awesome, and I'm absolutely excited about this new way of looking at data. What biological data is complex. It's very complex. And so what I've learned from my escapades in data analysis is that you've got to prevent yourself from trying to dive into it and looking at individual molecules, which is sort of where we came from in the deep, dark past. But rather you've got to sort of step out and just look for patterns. It's about pattern theory. Patterns, it might be two things that go up, two things that go down, and one thing that never changes. There's a pattern that might occur in that particular component of human beings that is predictive of some sort of behaviour. So we don't necessarily know yet what those patterns mean, but I think once we've got the patterns, then we can use them to dig deeper into the biology. But first, don't zoom in, but step out and look for interesting patterns. And that's something I've learnt is very powerful. I think data science is the new frontier. You know, I think we've seen this in the last decade or so in the context of genomics. You know, this has been absolutely fantastic. But now we've got other technologies coming on stream with other forms of omics. And the challenge really is to start to integrate multiple omics together to sort of build up a picture of how the human body works. And I think what we're going to find is more and more people want to get into this. And what I'm most invigorated by is the young kids. The young kids smell something really exciting and they want to be involved. And I want to help them be involved. I think if I had to give advice to a young student coming up through the ranks, so in other words, if I think back to when I was a young student, what would be going through my head? I think the future most successful researcher is somebody who can work at the boundaries between multiple disciplines within the one body. I think in the past what we've had is a situation where you'd have a biologist who collaborates with a mathematician. They work together, and that's been great. But I really think that the most successful future young scientist is going to be somebody who can do both of those things in the same skin. For some reason, it's been educated out of us. We were taught we should siloify ourselves. So I think we've got to now de-siloify ourselves, and young people have to embrace different things like math and biology or engineering and physics and so on and so forth. And that is going to be the successful young mind of the future, in my opinion.